Children, in the last class we had seen about in the biotechnology we had seen how you have to go for gene resistance and how the recombinant DNA transfer takes place. We had seen. Now today we will see polymerase chain reaction, that is PCR. PCR stands for polymerase chain reaction. Why do we need PCR is that we know that we can go for DNA cloning and recombinant DNA technology, but in the polymerase chain reaction what is happening, it takes place in 3 steps. That is, it takes place in 3 steps: denaturation, annealing and extension. As we can see from the name itself, it is polymerase chain reaction. That means an enzyme is required. That is, a chain reaction means it will be produced continuously. So what is happening here is denaturation. We will take a DNA strand. Suppose this is the DNA strand. This DNA strand is denatured, and we know that DNA is a protein. So protein means when it will get denatured, protein gets denatured on heating. So these 2 DNA strands heating, it should be exposed to heat, at least 92 degrees centigrade heat should be produced. So when the 92 degree heat, it is exposed to 92 degree centigrade, so it gets denatured. Denatured means these 2 strands have separated. Of the 2 DNA, the strands will be separated. Here, what we are using here for that, this is the DNA polymerase used here is known as Taq polymerase. Okay, first denaturing will take place, and Taq polymerase is used. It is not used just for separating of the 2 DNA strands, it is used for the addition also, for the RNA primers, small oligonucleotides. Okay, so for this strand also and for this strand also. So Taq polymerase, it's an enzyme, it is a DNA polymerase. We know what is the function of DNA polymerase. DNA polymerase adds nucleotides. It adds nucleotides. What kind of nucleotides? Oligonucleotides. Oligonucleotides are 5 to 10 DNA bases. So a short stretch of DNA primer. Okay, that is with the help of DNA polymerase, that is Taq polymerase. It is extracted from the bacteria Thermus aquaticus. It is extracted from this bacteria Thermus aquaticus because it can withstand high temperatures. So this DNA primer, okay, so this DNA primer that is also an oligonucleotide, 8 to 10 base pairs, it is added on either side of the strand, and this, after denaturation, this which is adding is known as annealing. Adding is annealing. This is added to either of the strands. After that, what is happening? This is a DNA primer. Obviously, it will extend. So the third point is extension. So we are getting a fresh strand. This was a DNA primer which was complementary to this DNA strand. This is a primer, 5 to 10 bases of oligonucleotide. This is complementary to this DNA strand. So we have produced a new strand. This new strand took place because it's a DNA primer. It will start reading itself. So extension. That means it has extended. It has produced a copy. Means earlier there were 2 DNA strands. You can see that it has replicated, and now we are having 4 DNA strands. Likewise, it will go on amplifying. This can go on amplifying. At least a billion times it can be amplified. So that is polymerase chain reaction, means chain reaction. It will go on 2, 4, 6, 8 like that. It will go on extending. So that way we can get such a big amplified copy. This can also be used. Other than that, what can we do with the copy which we have got, the desired DNA? This DNA can again be used for in a vector, and the cloned DNA multiple copies also can be produced. So PCR takes place in 3 steps, that is denaturation, annealing and extension. Okay. So this is the 3 steps how PCR takes place. After that, when we have got our desired DNA, be it through PCR or be it through recombinant DNA technology, we have got our desired DNA, desired gene of interest, desired enzymes. Now this enzyme or this protein, it has to be produced in so much quantity, large quantity we need. So for that we need big fermentation tanks. After that, we need this we have prepared. After that, it has to be produced in a large amount. How can we produce in a large amount is that there is two types of reactors which is mentioned in your book also, that is simple stirred tank bioreactor and sparge stirred tank bioreactor. Okay, so sparge stirred tank bioreactor. You are seeing in the simple stirred bioreactor, see how it is there, a motor is there, that is it is controlling this impeller and the foam breaker. So everything is being kept inside this, and various inoculums is also added to it. Sterile air is passed through the below of the tank reactor. Here acid base for pH control is being done, and from here steam, because it has to be maintained in a sterilized condition. So steam is being continuously fed for sterilization purposes. And this impeller, the function of the impeller is that it does not let the formation of bubbles. So if bubbles are not formed, surface area will be reduced. So that means there will be more, you will have space, because if bubbles are there, surface areas, they are occupying the area. The impeller, what they are doing is that they are breaking the bubbles, so that there is more space. So this is so big tank bioreactor. Here the enzymes, proteins, antibiotics likewise are produced. This is one simple stirred tank bioreactor. Other is the sparge stirred tank bioreactor. Here you can see this is the simplest one, because here bubbles are there. These bubbles increase the surface area, and for this the oxygen also surface area, it is being modified so that it will increase the surface area for oxygen also. This is the sparge stirred tank bioreactor. Mostly we are using simple stirred tank bioreactor, because everything is being maintained, and even the timer is also there, means for how much long, for how many hours we have to run, so that we get our desired product. Okay, so this is how we are getting a desired product. Tank bioreactor we are producing in so much quantity. After that, when means various antibiotic has been produced, proteins, enzymes we have produced, but after that it has to be tested, it has to be given a particular color, preservatives need to be added. So these things which are being done, that is known as downstream processing. So that is known as downstream processing. Basically what all we are getting from here to tank reactor, after that it should be tested. It should be tested on animals, or basically it has to be gone for testing in the labs also for its safety measures. Then various, what are the components, if you see in your medicine bottles also, you can see in the medicine packets also, you are seeing behind it is given how much quantity of which chemical is being added, and a particular color is also being, if you see the syrups, they have different colors, some is pink, some is orange, some is yellow. A particular color is also being given. Which type of color is being given synthetically by which chemical, that is also being shown, and that is mentioned in the bottle. Okay, and how much quantity of which chemical is used, that should also be mentioned in the bottle. Apart from that, its manufacturing date and expiry date, that also needs to be packed. And you see that many of the bottles, they are not white, they are colored bottles, so that if exposure to sunlight is there, they should not react. So you see some of the bottles are colored. So these are all the processes which go for downstream processing, means for safety, all the things should be mentioned, what is there, which are the components which are required for the making of this particular antibiotic, enzymes or proteins, for that downstream processing. After that it is being marketed, means when all the aspects are being seen, for its safety, for its color, for its components, everything is being seen, for tested on animals, everything is done, then it is being processed and it is being seen in the markets. Before that, so many processes have to be followed. So those all processes are known as downstream processing. Now we had seen, so this finishes the first chapter, all the topics have been finished, is the first chapter, biotechnology, principles and processes. In the next chapter, biotechnology in applications, we had already seen, I had taken that chapter because there was some connection with this chapter. Some of the topics were connected with the first chapter. So we had seen the gene therapy and how the insulin production was done, how hepatitis vaccine, how it is being prepared, stem cell technology, that was also being mentioned. Okay, so again in that chapter, that is applications in biotechnology, applications in biotechnology. What we will see in applications of biotechnology, we had already seen how it is genetic engineering is being done and how it is being used for the production of vaccines and how it is being tested on animals and how it is being done. So we will see what are GMOs, genetically modified organisms. We had seen that genetically modified organisms. Apart from that, what we will see that why these genetically modified organisms or genetically modified crops are used, why they are used. It is used because there are so many factors, at least 4 or 5 factors are there. First is that they help to withstand heat, cold, drought, flood. Okay, then they help to reduce post-harvest losses. Post-harvest losses means why we are using genetically modified plants or genetically modified organisms, why we are using. Third, in here we are mentioning is plants. Why we are using genetically modified plants, organisms in terms of plants, means disease resistance in plants, why it is being used, why genetic engineering is being used in plants. So these are the factors why we need biotechnology in the agriculture field. Why we need genetically engineered crops, why do we need. Then the third is that it helps to set against use of herbicides, pesticides. Okay, so it helps to that. Then one more thing is that enhance productivity, then disease resistance against various pests. Okay, so there are so many ways, so many at least 5 we are having such reasons that why we need this plants. Why the need of plants is that in the earlier class we had seen that how we are using agrobacterium tumefaciens, how we are using TI plasmids and how it is being introduced in the plants. So why do we need? These are the reasons why do we need. Because using these reasons we need so much facilities so that the crop production should be increased and it should be disease free. So for that what we are doing, we had used Bt toxin. What is Bt toxin? It is being isolated from the bacteria Bacillus thuringiensis. Okay, so we had learned about Bt cotton, Bt brinjal. What is Bt cotton and what is Bt brinjal? What is it that the gene which is isolated from Bacillus thuringiensis, it is being coded, and that as a TI plasmid it is being produced, that cotton plant which is produced Bt cotton, that is resistant to the cotton bollworms. Means if the cotton bollworm is injecting a leaf or the cotton bollworm it is injecting, so it releases a protein. That protein, what it does, it is like a crystal, it makes a crystal in the gut of the insect. It's an insecticidal protein. So it gets a crystal and the organism or the insect, it dies due to it. Similarly the Bt brinjal, because we need not spray any pesticides or any herbicide along with it, because it is being made such and it is insect specific. Means this Bt crops, Bt cotton, Bt brinjal, it targets only the specific organism, not all the organism. That way what is happening is we are maintaining the ecological balance also, in terms that we are not eliminating in a wide spectrum. We are only targeting a specific organism, and that too the brinjal also It is not sprayed with any weedicide or any pesticide. So the brinjal means it is totally free of pesticides, and it is itself capable of destroying the insect which feeds upon its leaves, its fruits. So it is a narrow spectrum. Okay, it is a narrow spectrum. So, but it has got its drawbacks also. BT cotton, BT brinjal, the seeds are very expensive, and in the long run means because it targets only a narrow spectrum of insecticides. We know that in nature we get resistant to this things. So what will happen, slowly what will happen, that it can develop resistance, means the insect also can develop resistance. Still now it has not developed resistance, it is targeting only that particular insect, and it is able to kill that particular insect, but the seeds are very expensive. So that's why if it needs to be produced, it needs to be produced in a very large scale, and that too it's very costly. That's why the farmers don't prefer BT cotton and brinjal. If it is if they get it at a subsidized rate, naturally they will go for it, because it is quite good in view of that it is not using pesticides for its growth. Okay, then another what we are seeing, that the cry gene. Okay, from the cry gene, this is the gene cry gene. From it is various toxins are isolated: cry A1C, cry 1 AB, cry 2 AC. So these are all the means the gene is common, the cry gene, from which the various toxoids or the toxins are being isolated. Children, we had seen that in the GMO, that is genetically modified organisms in plants, what we had mentioning about the cry gene and all. Apart from that, various 5 things which I had mentioned, it also increases the mineral usage. The genetically modified plants, the mineral usage is also increased of the plant, and biofortification. What is biofortification is that we have seen that in the ration rice, what you are seeing, that you are getting carbohydrate, rice, atta, wheat, what we are getting, you are getting carbohydrates from that. But what we are seeing it in India, it's a developing country, and most of the people are BPL, that is below poverty line. So what you are getting in the ration is only rice. In some places you are getting only rice. But what if that rice is being enriched, means only, it is not that it is only providing carbohydrates, apart from that it is being increased, means it is providing an additional thing which will be useful for the child or the family. Because majority of, in India what is happening, majority of the children are suffering from xerophthalmia, that is childhood blindness. Okay, nowadays it has reduced. Why? Because people are aware of it, aware of xerophthalmia, childhood blindness, because in the various Anganwadis also food is provided which is a balanced diet, so that the children, if the parents who are not able to afford, so they can send their children to Anganwadi. In Anganwadi provisions are also being made so that the child is being fed the balanced diet. Means even if khichdi is being provided to the child is getting khichdi, means the child is getting rice which is rich in carbohydrate, dal which is rich in protein. Apart from that, that khichdi is being enhanced with some carrot or some vegetables, some even the locally available various green vegetables which are added in that khichdi, so that the child is not devoid of vitamin A deficiency. So that is because many people are not following that truly. Even the Anganwadi workers also, we are not seeing that every time they would be adding vegetables. Could it be that they could only give the rice and dal mixture as a khichdi for the children? So what we can do is, what the scientists are doing is that they are going for biofortification, means the rice is prepared is known as the golden rice. Why it is known as the golden rice? Because it is enriched with vitamin A. It is enriched with vitamin A. So if at all the child is only taking rice also, the child is having carbohydrate enriched with vitamin A. So we will not get children suffering from xerophthalmia. It has reduced. Earlier there were so many cases of xerophthalmia, but nowadays due to the awareness and by the government also, they are providing so much facility and there is so much awareness. Even in the Anganwadi workers also, they are able to say what is a balanced diet, what does the child need. They are so much aware, and thence we are able to curb this xerophthalmia. We are not able to totally curb, but to a very large extent it has been reduced. Okay, that is golden rice. That is biofortification. With this, if suppose we are adding in other foodstuffs also, in the this also we can add vegetables, in the fruits what we are getting, it can be a biofortified fruit, it can be a biofortified vegetable also. So that can be used, and that can be done in the laboratory itself, and it is being done and it is being followed. So what will happen, means we are getting an additional thing, means if you get 1 + 1 free, you are so happy. So suppose you are getting carbohydrate, apart from carbohydrate you are getting mineral also, you are getting vitamin also. So it's an added benefit to the person or the individual, because at the same rate you are getting a amplified version. Okay. So that is biofortification. So what we had seen is that Bt toxin, which is being Bt toxin which was being isolated from the Bacillus thuringiensis, from the gene Cryogen. From this gene, this is being made. So there are so many Bt toxins, Bt Bacillus thuringiensis toxin, which are controlling. They are insect specific, like I said earlier also, Cry A1C, Cry A1B, Cry 2AC. Okay. Apart from that, there is also a thing known as RNA interference, means that is present in the nature itself. RNA interference. Okay. RNA interference. What is happening is, in the RNA interference, what is happening, there are two types of enzymes. One such enzyme is known as the dicer. What is happening, the RNA, which is a double stranded RNA, suppose this is a double stranded RNA. Okay, I am mentioning in plants, RNA interfering in plants, I am mentioning. Okay, so this, what is happening, this double stranded RNA, with the help of this dicer enzymes, it will cut this into small bits. Before that, I want to say, what is happening is that, what is the function of an RNA? Means it helps in the translation process. Means DNA to RNA, that is transcription. RNA to protein is translation. So what is happening in the RNA interference? RNA interference is not making the production of a protein, means it is hampering in the production of the protein. It is not making the protein, that is the protein is being hampered. So what is happening is that the dicer, this enzyme, it will, when it is inserted in the plant cell, what is happening in the RNA, it breaks down the RNA into small fragments. These RNA, these small bits of RNA, the dice RNA, this is being taken by the nematode. This is being taken by the nematode. And when these small fragments are added, taken by the nematode, what happens when the translation process takes place in the nematode? This is also being there, small RNA, this is also there. So when the translation takes place in the nematode, this, what will happen, this complete RNA, this is broken down into small strands. It will break down, means the dicer, when it will attach, when the nematode will take this small dicer RNA, the nematode is injecting from the plants, this dicer, it goes through the nematode, and when the nematode is performing the process of translation in its own body, what is happening, this will cut, means see, if this mRNA, if this strand is regular, then only a protein will be produced. But if there is a hindrance, means if there is a breakage at various points, so there will be no protein synthesis. That is what is happening. See, it is breaking, these strands are being broken down. So it is not continuing translation, the translation process is being stopped. The translation process is stopped, means the protein is being, formation of protein is being stopped. So that is what is happening in nature, it is happening, that is known as the RNA interference. RNA interference means RNA interferes with the formation of protein, that means it stops the translation process, and this is what is known as the RNA interference. It happens in plants. Now next we will see, we have seen RNA interference, we have seen the Cryogen. Okay, so basically we are seeing that we saw it in plants. Now what are transgenic animals? What are transgenic animals? Transgenic animals. Transgenic animals means transgenic, their genetic manipulation. Genetic manipulation means their DNA is altered ऑल्टर्ड मतलब थोड़ा उसको ठीक करेंगे, मैनिपुलेट करेंगे, थोड़ा जैसा कुछ चेंजेस लाएंगे डीएनए में। तो ट्रांसजेनिक एनिमल्स वो एनिमल्स होते हैं कि जिनका जीन मैनिपुलेट किया जाता है, मतलब दे आर हैविंग अ फॉरेन जीन इन देम, एंड दैट फॉरेन जीन, with that foreign gene they are being modified, that with that foreign gene they are being produced, they are being modified, वो foreign gene के साथ ही वो आगे बढ़ते हैं। ट्रांसजेनिक एनिमल, दे हैव इन देम द फॉरेन जीन, okay, so because that DNA is being manipulated, that they are having a foreign DNA in them, थोड़ा या foreign gene in them, जो हमने खुद डाला है उसमें, मैनिपुलेट करके डाला है। तो such organisms are known as, और such animals are known as ट्रांसजेनिक एनिमल्स। लेकिन हम ऐसे एनिमल्स बनाते क्यों है? वाई डू वी रिक्वायर ट्रांसजेनिक एनिमल्स? क्यों इनका डीएनए ऑल्टर किया जाता है, क्यों हम बना रहे हैं उनको? फर्स्ट, नॉर्मल फिजियोलॉजी, फॉर नॉर्मल फिजियोलॉजी। सेकेंड, स्टडी ऑफ डिजीज, स्टडी ऑफ डिजीज, ओके। देन बायोलॉजिकल प्रोडक्ट्स, biological products. Then fourth, vaccine safety, vaccine safety. Fifth, chemical safety tests. So for the normal physiology means in humans also means for all the organs to work properly, if there is any problem in any organ, that needs to be studied and tested. So every time a human cannot be taken na for testing. So what is happening? The transgenic animal, transgenic mice, pig, these all are being taken, mostly mice, for their physiological functions, means how physiologically how their systems are working, and if this chemical is being added, how more if it is functioning properly, means if there is any defect, how it will function. Second, study of disease, like we did in insulin, how it was being done. So every disease, if we are going for it, means how the human growth, like we had seen normal physiology, how the growth should take place. Here for various study of diseases, cancer, hepatitis, anything, okay, AIDS also, the study is being done, means it will be done on an animal. So for that, transgenic mice, basically we are using transgenic mice and all for the study of various diseases. Third is the biological products. We need the biological products, so that will be administered first in the animals, and then if it is being found ki okay they can use, then we can also use them. Fourth is the vaccine safety. Any vaccine which is prepared in the industries and that is being tested prior to a human, prior to on humans, it is a trial is being made on animals, so that it is being tested, and if the animal survives, means that means it is safe for the humans also, then only it will be administered in the markets to humans, it will be manufactured. Last, chemical safety test. Now also we are seeing that various chemical trials are being done for the COVID test also. First it is done on the animals, then if the animals are going for a thumbs up, means they are fine with that vaccine, then it is being tested on humans, the trials which had been done. It was priorly, it will be done on animals, then only the humans will be exposed to such vaccines. Okay, those are the transgenic vaccines. Apart from that, what we had seen is that there is so much wealth, traditional knowledge and the resources in India. Like in the forest also we are having so many medicinal plants, okay, so many medicinal plants, and that traditional knowledge, so many traditional knowledge we have. Okay, what happened in the 90s, 1990, 1995, 1997, what was happening, what all was our traditional knowledge, the patents. Patent means it is a right, they registered it in the European Federation, okay, product organization, EPO, European Product Organization. So there they will register that this is being found by me, this plant is being found by me, and all the alkaloids, or which are all the things which are used for various studies, which is beneficial for such and such diseases, that I know, and I am the sole founder for this plant, and I need a patent for that plant. So if they EPO, if they gives a patent, that means in future if any child wants to do any research or any medicine has to be prepared, we have to give a certain amount to that person who has the patent for it. So what happened in the 1990s, US companies, many of the US companies, they took patent for neem, they took patent for turmeric, which was our Indian traditional knowledge and medicines. So there was a huge fight for that, and we somehow in the 1997 also we got the rights for neem also and haldi also, that is turmeric, we got the rights for that, but it was a huge battle. Ultimately it was known that we should have our property rights, that patent rights should be there. That's why after so long patent bill was passed in India. In 1997, that was about haldi and neem, and much widely publicized was in 1997. It was by a Texas based company. They used the term basmati rice lines, okay, basmati grain lines, basmati rice lines, they used by this, and they took the patent for basmati rice, which is we are the sole owners for basmati, India. So they had taken a patent for basmati. That means if any of our agricultural scientists are doing any of the research in basmati to prepare a much longer grain and a much good flavor, means we have to give certain amount to that US based, Texas based company, okay. So they gave the name basmati. So there was a huge legal battle, and in 2002 we won, and that basmati, that name was removed from the Texas based company, and it was known as the grains 867 grains lines, it was replaced. Okay, so what I want to say is that this is biopatent. This is biopatent. So we should take patent. Otherwise, what is happening? This which took place, the complete thing is known as biopiracy. Piracy means chori. They had taken it from us. They had taken a thing which belonged to us without our knowledge, and they were minting huge money due to biopiracy. So this biopiracy needs to be stopped. So how could it be stopped? As and when we are finding a new plants, any knowledge based on it, we could go for a patent, biopatent. In India itself, patent is being, we should do. Before they realize that, we should take a patent for that. And what are all our traditional knowledge, the farmers who know about that plant and all, they should not be deprived of that knowledge. They should get some amount of that. Instead of that patent going to some other country, we should see that the poor farmers or the persons who have the traditional knowledge of these medicines, who had at first knowledge, hands-on knowledge they gave on that particular plant, they should know about that plant and they should get benefit of that amount. So for that reason, biopatent should be done. So prior to that, what I did, what we had mentioned is that in the transgenic animals also, why they had been used. So there is a committee, that is Genetic Engineering Approval Committee, okay. So there is a committee, that is Genetic Engineering Approval Committee, Genetic Engineering Approval Committee. So what are findings we are doing? What all findings in terms of what we are extracting from the animals, or what we are doing on the animals also, what we are doing on the plants, that will be passed through this committee, that is Genetic Engineering Approval Committee. After the Genetic Engineering Approval Committee, it goes for all the factors, ethical factors, various moral factors, and these all factors, okay, they will be seen, and if all are going right, then only that will be passed, the product will be passed. Okay, then you can continue with this product, it has got no problem. So various things needs to be kept in mind when we are doing a genetic engineering experiment, be it on animals or be it on plants. There is so many rules and all, and it should be done very properly so that the result should be beneficial to the society. Thank you children, stay home, stay safe.